Imagine if going to the gym didn't just build your muscle, but actually forced your muscle cells to physically travel back in time to when you were an infant.
Right, which sounds completely like science fiction.
It really does. But today's deep dive reveals that this is, almost exactly what happens when we zap the human brain with electricity to cure severe depression.
Yeah, it is a profound shift in how we understand the brain's capacity to heal. I mean, for decades, the medical field has relied on treatments like electroconvulsive therapy, ECT, and repetitive transcranial magnetic stimulation, or RTMS.
Where we are literally applying electrical currents or strong magnetic pulses directly to the brain.
Exactly, to treat the most intractable, severe cases of mental illness.
And the brutal reality is that we do it because it works. Like when traditional antidepressants fail, when talk therapy isn't enough, brain stimulation can be incredibly effective. It literally saves lives.
Does. But for all that success, psychiatry has been operating with this glaring blind spot.
Right, the black box.
Yeah, we've known the macro effect that the patient's depression lifts, but the micromechanism, the actual cellular biology of how electricity fixes a mood disorder, has been a complete mystery.
We've basically been hitting the side of the television to fix the static on the screen without knowing, which wires inside were actually being reconnected.
That's a great way to put it, but the sources we're looking at today change that entirely.
Yeah, we are pulling from a stack of recent research for you guys today, but the crown jewel is this groundbreaking 2026 paper published in Nature Communications by Murano and a brilliant team of researchers. The paper is titled Repetitive Neuronal Activation Regulates Cellular Maturation State via Nuclear Reprogramming.
Which is really just the academic way of saying they have finally mapped out the exact molecular domino effect that happens inside a single brain cell when it's stimulated.
So the mission for this deep dive is to open up that black box. We're going to explore how zapping the brain physically rewinds the biological clock on mature neurons, the bizarre trade-offs that come with this cellular time travel, and what that means for the future of mental health treatment. Okay, let's unpack this.
Well, to really isolate what electricity does to a human brain, you run into a massive logistical problem right out of the gate.
Because you can't exactly open up a living person's skull during an ECT session to watch their neurons fire.
Right, you cannot easily study this in humans, and even if you magically could, a full brain electrical seizure is incredibly messy.
It would be like trying to isolate the sound of a single violin while sitting in the middle of a chaotic, blaring symphony, right? There's just too much noise to track cause and effect.
That is the exact hurdle that kept this a mystery for nearly a century. So the researchers in this study bypassed the human brain entirely to start. They turned to optogenetics in mice.
Oh, optogenetics is fascinating.
It really is. It's this revolutionary technique where scientists genetically engineer specific, targeted cells to become highly sensitive to light.
And in this specific study, the source material notes they used a genetically modified line called POMC-CRUI, CHR2EYFP mice.
Quite the alphabet soup.
Yeah, let's decode that for a second. They essentially engineered these mice so that only one very specific type of cell would react to blue light. They targeted the granule cells in the dentate gyrus.
The dentate gyrus is the perfect target for this, by the way.
Yeah.
It's a specialized region tucked inside the hippocampus, which is, the brain's hub for learning, memory, and emotional regulation.
Right.
By packing these specific granule cells with a light-sensitive protein called channel rhodopsin 2, they could use tiny implanted optic fibers to shoot blue light into the brain and trigger only those neurons to fire.
So there's no messy electrical storm, no full brain seizure.
Exactly. Just pinpoint laser-focused activation.
And they called this highly controlled protocol REPOPI test repetitive optogenetic stimulation.
Right, and the dosage is key here. The researchers delivered 10 millisecond light pulses at a frequency of 10 hertz for just 5 minutes a day.
That is an incredibly brief window, just 5 minutes of targeted brain activation.
But they set up a really fascinating comparison. They measured the brain changes in mice who got this REPOPI treatment for 3 days versus mice who underwent the protocol for 10 days.
And the difference is huge, right?
The contrast is staggering. In the mice that only received 3 days of stimulation, there were noticeable changes in the brain cells, but they were entirely fleeting.
Like they just went away.
Yeah, within 2 weeks, those neurons had completely reverted back to their normal baseline state. It was just a temporary blip. However, the 10-day stimulation caused massive fundamental structural changes in the brain cells,
Wow.
and those changes were stable, lasting for well over a month.
It makes me think of the difference between going to the gym once versus surviving a grueling 10-day training camp.
Oh, that's a good analogy.
Because that single gym session makes you sore for a couple of days and then you're right back to normal. But the sustained stress of a 10-day camp actually changes your resting metabolism, right?
Oh.
It alters your muscle architecture for the long haul.
Yeah, you need that cumulative load to force a biological adaptation.
But does that 10-day threshold in mice actually translate to how we treat humans? Like, does the timeline match up?
What's fascinating here is that this perfectly mirrors clinical ECT.
Wait, really?
Yeah. When a patient undergoes electroconvulsive therapy for severe treatment-resistant depression, a single session almost never provides lasting relief. The standard scientifically validated clinical protocol typically requires a patient to undergo a course of 6 to 12 sessions over several weeks to achieve a durable outcome.
That's incredible.
The fact that this 10-day tipping point in the mouse brain maps so cleanly onto the human therapeutic window, it heavily suggests a shared underlying biological mechanism.
Which brings us to the core mystery of the whole deep dive. What is that mechanism? What actually happens to these granule cells after those 10 days of stimulation?
Well, you would assume, based on how we usually talk about neuroplasticity, that the cells just get stronger or they build more synaptic connections.
Right, they level up.
But the paper shows they actually get younger. They regress.
They lose their mature identity entirely.
Yes. To measure this objectively, the researchers tracked a specific protein called calbindin. In the dentate gyrus, calbindin acts as like a biological badge of maturity.
Okay.
If a neuron is fully developed and functioning like an adult, it expresses high levels of calbindin. But after 10 days of a repop-PS, the levels of calbindin in these targeted cells absolutely plummeted.
So the cells were literally stripping off their markers of maturity.
Exactly. But they took it a massive step further than just tracking one protein. They performed RNA sequencing on these cells.
And for those listening, that involves reading the entire active instruction manual the cell is using at that exact moment.
Right. And the gene expression profiles of these adult mouse neurons morphed so drastically that they no longer looked like adult cells at all. They closely resembled the brain cells of an 8-day-old infant mouse.
Which is wild.
The researchers termed this a pseudo-immature state. The cells essentially hit a biological rewind button.
But, studying a mouse's reaction to a laser is one thing. To prove this applies to the devastating reality of human depression, they had to look at human brains, right?
They did. They cross-referenced this mouse data with human RNA sequencing data. They examined post-mortem brain tissue from human patients with severe mood disorders who had successfully undergone ECT before they passed away.
Well, if this theory holds up, those human brains shouldn't look like mature adult brains at all, right? The data should show the exact same regression toward infancy that we saw in the mice.
That is the exact one-to-one correlation they found.
Oh, wow.
The genetic shift toward immaturity in the mouse brains matched the genetic shift in the human ECT patients flawlessly.
That's amazing.
In both human and mouse brains, the genes responsible for coding AMPA receptors, specifically the GRIA1 and GRIA2 genes, dropped significantly.
Let's contextualize those AMPA receptors for you listening. Think of AMPA receptors as the heavy-duty communication satellites that mature neurons use to send complex signals to each other.
That's exactly what they do.
So when a cell regresses, it basically takes those satellites offline. It stops trying to communicate like an established adult.
And at the exact same time those communication satellites went offline, the expression of a completely different gene called ARC skyrocketed.
ARC.
Right? ARC is heavily involved in synaptic plasticity. So by shutting down the mature AMPA receptors and cranking up ARC, the cell forces itself into this highly flexible, moldable, immature state.
I really want to emphasize the sheer scale of this discovery. We are talking about electricity in humans or, targeted light in mice, forcing a mature, set-in-its-ways adult cell to completely de-differentiate.
Yeah, it's literal cellular time travel. It's known as nuclear reprogramming.
But we have to ask how. How is a brain cell physically executing this massive regression without just destroying itself?
Well, this leads us to arguably the most paradigm-shifting genetic discovery in the paper.
Okay.
When the team analyzed the RNA sequencing data, they found incredibly high expression of G2M cell cycle genes.
The G2M phase.
Yes. These are genes like cyclin B1, B2, A2, and CDC25C.
Wait a second. I have to stop you there and push back.
Go for it.
I remember high school biology pretty clearly, and I thought adult brain cells don't divide. They are postmitotic.
That's what we're taught, yeah.
Right. Once your brain develops them, they are locked in. The G2M phase is the stage of the cell cycle right before a cell physically splits into two. How can a mature neuron be entering the G2M phase of cell division?
It completely violates a core assumption of neurobiology. But you are correct. The neurons aren't actually dividing into two new cells.
So what are they doing?
They are re-entering the cell cycle, turning on all the heavy machinery required for division, but then they stall out. They hit the brakes right before the cell actually splits.
Oh, I see. It's like packing all your belongings into cardboard boxes because you're getting ready to move to a new house. But then you just stay in the apartment living out of the boxes. Like your entire living structure is completely remodeled and chaotic, but your physical location hasn't changed.
That analogy captures the mechanics beautifully. The cell is remodeling its entire 3D genome structure without tearing itself apart to divide.
And they could see this happening.
The researchers actually witnessed this happening inside the nucleus. The nuclear lamina, specifically a structural protein called lamin B1, which acts like a protective cage around the DNA, it became physically disrupted.
So the cage breaks open.
Yeah. And the DNA itself, the chromatin, condensed into these dense clusters called chromocenters. They even saw a huge spike in phosphohistone H3, which is, the classic textbook marker that a cell is actively in mitosis.
And the trigger for all this remodeling starts with the light or the electricity.
Exactly. The stimulation causes a massive influx of calcium into the cell. That flood of calcium activates specific kinase enzymes, which act like molecular messengers, and those messengers flip the switch on that G2M zombie phase.
But they didn't just observe this, right? They engineered a way to prove it using CRISPR, which frankly blew my mind when I read it.
The experimental design here is just brilliant. To definitively prove that this stalled cell cycle phase was the actual cause of the brain's rejuvenation, they used a CRISPR-Cas9 gene editing system.
But getting CRISPR into a living brain is incredibly difficult.
It's notoriously hard, so they used an AAV9 viral vector.
Right, so essentially they hijacked a harmless virus, scooped out its original viral programming, and used it as a microscopic Trojan horse.
Yes.
They packed the CRISPR editing tool inside the virus to sneak it past the brain's security system.
And once inside those targeted neurons, the CRISPR system functioned like molecular scissors. It surgically knocked out the cyclin B gene.
And cyclin B is the master regulator that initiates that G2M cell division phase.
Precisely.
So without cyclin B, the cells can't even begin to pack up the boxes. They're completely locked out of that zombie division phase.
Which gave the researchers the ultimate control group. When they knocked out cyclin B and ran the entire 10-day RE-PAPS protocol on the mice again,
What happened?
absolutely nothing happened.
Wow.
The protective cage around the DNA didn't break down. The markers of maturity didn't drop. The dematuration process stopped completely. And most importantly, the behavioral improvements in the mice vanished.
So cyclin B was the definitive linchpin.
It was the linchpin.
Okay, this deep dive into the nuclear architecture of a cell is fascinating. But let's zoom out to the living, breathing mouse for a second.
Sure.
If we successfully force these cells into an infant brain state, how does that actually affect the daily life and behavior of the animal? Like, does it cure them?
The behavioral changes were undeniable. They put the mice through several standardized evaluations, including the tail suspension test and the novelty suppressed feeding test.
And for context, these are classic behavioral models used to measure depressive and anxiety-like states in rodents.
Right. In the tail suspension test, a depressed mouse will quickly give up and remain immobile, whereas a resilient mouse will keep struggling. The mice that received the 10-day or RE-PAPS protocol showed significantly reduced immobility. They fought harder.
That's a clear win.
Yeah, and they also showed less anxiety when introduced to new environments in the feeding test.
But it didn't just show up in stressful tests, right?
No, not at all. When the researchers monitored the mice in their normal home cages, their baseline locomotor activity, just their general energy, curiosity, and movement around the cage was significantly higher.
And the crazy part is that elevated energy lasted for over a month after the stimulation stopped.
Yes.
But if the stimulation only took 10 days, why does the behavioral cure last so long?
The longevity of the cure comes down to a specific transcription factor called delta FOSB.
Delta FOSB.
When the calcium flood triggers the cyclin B cascade, one of the downstream effects is an upregulation of delta FOSB. Now, most proteins in the brain degrade and vanish within a few hours.
Right, they're fleeting.
But delta FOSB is highly unusual. It has a biological half-life of roughly 208 hours.
I mean, that is an eternity in cellular time, over 8 straight days just for half of it to degrade.
And because it lasts so incredibly long, delta phosB acts as a molecular anchor. It essentially locks the newly remodeled cell into this immature, highly plastic state for upwards of 40 days, long after the physical stimulation is ended.
So it forces the window of neuroplasticity to stay wide open, giving the brain time to rewire its depressive circuits.
Exactly.
Here's where it gets really interesting, though. Biology rarely offers a free lunch, you know?
That is very true.
You can't just force a mature brain cell to mentally regress to infancy without paying a price. And the researchers identified that price using some incredible technology called Invoke miniature microscopes.
Oh, these are a marvel of modern neuroscience.
Yeah.
The Invoke microscopes are tiny, lightweight devices surgically mounted directly onto the heads of freely moving mice.
Just tiny little cameras on their heads.
Basically, yeah. They allow researchers to perform in vivo calcium imaging. This means they can literally watch individual neurons light up and fire in real time while the mouse runs around its environment.
And when they monitored how these newly rejuvenated dentate gyrus cells were processing information, they discovered a severe trade-off.
They did. In this part of the brain, we rely heavily on two types of neurons: speed cells, which encode how fast we are moving, and place cells, which act as like your internal GPS, creating a spatial map of where you are in a room.
And after the 10 days of stimulation, the speed cells became much sharper. The mice were moving more, and their brains were tightly tuned to that physical action.
Right. But the place cells performed significantly worse. The cells had lost their mature ability to map the environment accurately.
The GPS broke.
If we connect this to the bigger picture, this finding perfectly explains the dual nature of human ECT.
Oh, wow. Because of the movement.
Yes, the dramatic increase in speed cell activity correlates directly with the lifting of depressive immobility, that heavy, crushing paralysis that often defines severe clinical depression. The patient regains their drive to move and engage.
But the degradation of the place cells.
That explains the single most notorious feared side effect of ECT, anterograde and retrograde amnesia.
The memory loss. Patients often struggle to form new spatial memories or recall events from right around the time of their treatment.
Precisely because the cells responsible for mapping those memories have been forced into an infant state.
That makes so much sense.
The human brain cells are sacrificing their mature spatial mapping abilities in exchange for a systemic mood reset. The neurons quite literally have to forget their rigid, mature identity to become flexible enough to escape the depressive loop.
Which introduces a brilliant yet terrifying paradox that the paper addresses, this concept of the bistable switch.
Right.
We've just spent this entire deep dive establishing that making brain cells immature is the secret to curing depression. But neurobiology shows us that this exact same cellular immaturity is found in the brains of people suffering from severe epilepsy or, the devastating cognitive decline of Alzheimer's disease.
It is a paradox.
So why does the cellular amnesia heal one brain but completely destroy another?
It all comes down to the context of the stimulation. The researchers propose that this immature cellular state acts as a bistable switch. Think of it like a biological thermostat. It isn't inherently healing or inherently pathological. It depends entirely on how it is controlled.
So in therapies like clinical ECT or the REPA-POPS protocol with the lasers, the stimulation is brief, it's intermittent, and heavily regulated by doctors.
Exactly. You trigger the reset, force a brain stuck in a rigid depressive state to become flexible, and then you stop. You let the ecosystem heal.
But in devastating diseases like epilepsy or Alzheimer's, the brain is experiencing chronic, uncontrolled hyperexcitability.
The biological system is short-circuiting, and the stimulation never stops. Because the brain is constantly overactive, the cells get trapped in a permanent, pathological loop of immaturity.
Oh, so they keep hitting the reset button over and over.
Over and over, yeah. Meaning the cells can never mature back into functional, healthy neurons that hold memories.
That is tragic, but it makes so much sense biologically.
This raises an important question about the future of how we view psychiatric treatment. The boundary line between a life-saving intervention and a pathological disease state is incredibly thin.
It's entirely about the Goldilocks zone of neural activation. A targeted, rhythmic dose of electricity provides a desperately needed reset, but chronic, uncontrolled activation leads directly to neurodegeneration.
Yeah, it forces a total shift in how you think about your own brain. It is not a static hard drive where a doctor just needs to find the bad file and delete it.
It's a highly dynamic, flexible ecosystem. And this study proves that if you introduce the exact right rhythm of stimulation, you can force the whole ecosystem to tear itself down to the studs and rebuild.
And because we can now trace that entire rebuilding process down to a specific master gene, cyclin B, and a specific molecular anchor, delta FosB, we are finally moving the field of psychiatry out of the dark ages of trial and error.
We know exactly what the electricity is doing.
We do.
So what does this all mean?
We started this deep dive with a stack of highly complex neurobiology research, and what we uncovered is that repetitively stimulating the brain forces mature, set in their ways cells to regress into a youthful plastic state.
Right, they achieve this by hijacking the machinery of cell division.
Entering that zombie G2M phase that remodels their nucleus, which ultimately lifts the crushing immobility of depression at the temporary cost of memory and spatial awareness.
And thanks to the long lifespan of delta FosB, they hold on to that youthful flexibility for over a month, providing a critical window for the patient to establish healthier, non-depressive neural pathways. It is a breathtaking biological mechanism, but it leaves me with a thought that I want you, the listener, to mull over as you step away from this deep dive today.
I like where this is going.
Now that the mystery of the black box is solved, now that we know the exact molecular cascade that the calcium influx triggers cyclin B, which remodels the nucleus, which then triggers delta FosB to anchor the cell in that flexible state, could the future of psychiatry bypass the brute force of electricity entirely?
It is the next logical frontier for psychopharmacology, for sure.
What if we could develop a targeted pharmaceutical? A pill that crosses the blood-brain barrier and directly initiates this precise bisensible switch of nuclear reprogramming.
That would change everything.
Imagine a world where a patient can get all the life-saving, world-altering benefits of ECT, the rapid lifting of the deepest, most intractable depressions without enduring a single electrical shock and without sacrificing a single memory.
It's an incredible thought.
That diagnostic X-ray machine for mental health might not just be fixed, we might be on the verge of replacing it entirely. Thank you for joining us on this deep dive. Keep questioning, keep exploring the science, and we'll see you next time.